Now we're going to take a look how to perform protein gel electrophoresis using polyacrylamide gels. I have my sample of protein that I want to visualize. So the first thing I need to do is again add my loading dye. The loading dye is going to allow my sample to go into the gel. This is a 2x concentration, so here I have 25 microliters, so I'm going to add 12.5 of my loading dye, And you want to pipette up and down slowly because you do not want to introduce any air bubbles. To help denature the proteins, typically you take your protein sample and you add it to a hot water bath. Now we're ready to set up our gel. Here I'm using a pre-made gel that is 15% concentration. So I'll take it out of its case. And the gel is between two pieces of plastic or glass. The comb that allows for the wells to be formed can be lifted up as such, and it is these wells that we are going to load our protein samples. To load our gel into our gel box here, take this down, we lift up our electrodes, open up the clamps, and if you look at this gel, you'll notice that one of the pieces of plastic is shorter than the other. So this side is shorter than that side. You want to orient the gel such that the shorter end is towards the inside, like such. And then clasp it on. That then goes into our gel box here. Just like with the DNA agarose, I need a running buffer, and the running buffer that we use for proteins is tris, glycine, and SDS. So we'll open that up, and then we pour it into the box. And you want to make sure that the buffer is both on the outside as well as the inside. Just need a little bit more. This gel box is set that you can run 2 protein gels at a time. Since I'm only running 1 protein gel, I have just a piece of plastic on the other side in order to retain the running buffer inside of the electrodes here. So to load our sample, we'll use a micropipetter and these special gel loading tips. You'll notice that they have a much thinner shaft down at the tip end. So I'll take my protein sample. And remember, this is the protein sample that had the loading dye and was heated for several minutes. Certain gel electrophoresis assays involving proteins don't involve heating the sample. So make sure you check your lab protocol or with your lab professor if you are supposed to heat the sample before loading. Now to load, I take my micropipetter and my tip here, and I'm going to insert the tip between the 2 pieces of plastic. And I'm going to enter into my well here and slowly begin to push out the protein. You know if you're doing this right if you see the protein accumulating within the well. I then remove the tip, and as you can now see, my protein sample is loaded into the gel. Just like with the DNA gel electrophoresis, we had a ladder. So here we also need a ladder to determine the size of my proteins. And here I'm using a pre-stained molecular ruler. So I'll take a little bit of that out as well. This one already has the loading dye added. And I will go into the next well, again, between the two pieces of heavy plastic, so because that's where the gel is, and I'm going to slowly pipette that in. And again, I know I'm successful when I see the sample is stored in the well I just pipetted in. Once all your samples have been loaded, you then connect black with black, red with red again, as such. I'll plug this into my power source. Turn on the power, move this out of the way, and then let it run. So what will happen is, as our electric field is generated, the proteins are going to begin moving from the top of the gel all the way down. And you can see the formation of bubbles, and just like with our DNA gel, the presence of the bubbles show that you have established your electric field. So we're going to let this run until the bands go all the way down the gel and get close to the bottom of the gel. Let's imagine now that our sample has resolved and has gone all the way down to the bottom. So the next thing we have to do is to take out the gel and put it into stain. So I'm going to stop the electricity. I open up my box, remove my sample, and pour off the extra liquid. Again, for the purposes of our demonstration, you would wait until the loading dye has reached the bottom of the gel. But I'm going to open up the clamps and take out my gel. So again, the gel is between these two pieces of, it can either be glass or plastic. So in order to get to the gel itself, you can take a spatula and pry open the cast, like such. So here's my open cast now. You can see that the gel is in between. I can remove my gel and put it into stain solution. This is a stain that is known as Kumasi blue. We put the gel into the Kumasi blue. We close the container. This Kumasi blue solution contains the stain Kumasi blue, acetic acid, and methanol. You let this rock until you begin to see the bands of your protein, in which case you then remove the stain or put the gel into a de-stained solution, which just contains acetic acid and methanol. You can keep rinsing with that de-stained solution until you see clear bands present.